Hello, I'm Mariam Faruqi and I'm part of Team 6 in the Fire Bacterial Pathogenesis Lab. Our lab is focused on the bacteria Pseudomonas aeruginosa, which is a common cause of hospital-acquired infections, or HAIs. The HAI our lab is interested in is catheter-acquired urinary tract infections, or CAUTI. CAUTI happens when bacteria contaminates and forms biofilm on the catheter that is inserted in a patient and spreads to the tissues and organs in the urinary tract. Biofilms are a complex matrix of bacterial cells and secretion products that form on different surfaces when in an environment that promotes their growth. Bacteria within a biofilm can be released and move through the catheter to infect the patient's urethra and bladder, leading to CAUTI. If left untreated, the infection can spread to the kidney, leading to kidney failure, sepsis, and death. Pseudomonas aeruginosa is a common cause of CAUTI. However, how Pseudomonas aeruginosa causes CAUTI is not well understood. Our team focused on a specific gene in Pseudomonas aeruginosa, PA14-630-80, or LLDP, which was previously shown to be overexpressed when the bacteria was grown in LB media and 0.5 molar urea. Our research project's aim is to find out the connection between LLDP and biofilm formation, and why it was overexpressed in LB media and 0.5 molar urea. LLDP encodes the protein L-lactate permease, which is a lactate transport protein. Through scholarly research, we found that lactate has been found to contribute to biofilm formation in other bacterial species, E. coli and Bacillus subtilis. Since lactate is found to be present in urine, it can be rationalized that during the infection, the increase in lactate available for the bacteria will lead to an uptake in the lactate transport by the PA14-630-80 gene, which would lead to an overexpression of this gene when the bacteria is grown in urine. Our hypothesis was that if LLDP expression is reduced, then there would be a loss of nutrients for Pseudomonas aeruginosa, and it would not be able to survive, which would then lead to little or no biofilm formation, which is essential for Pseudomonas aeruginosa CAUTI. To test our hypothesis, we will compare biofilm formation in a Pseudomonas aeruginosa strain that has reduced expression of LLDP compared to the parental wild-type strain. First, we would knock down the expression of LLDP by using the CRISPR-Cas9 molecular tool. Then we would make sure that the knockdown worked by testing if the bacteria with reduced expression is able to grow in media with only lactate as a carbon source. If it doesn't grow, it shows that the knockdown worked. Then we would grow the LLDP knockdown strain and parental wild-type strain in an LB media plus 0.5 molar urea media and compare the biofilm formation using a crystal violet dye assay. Since we didn't perform this experiment, the data we have here is mock data for our expected and alternate results. And if our results show that the LLDP knockdown strain has less biofilm growth than the parental strain, we can conclude that our gene PA14-630-80 contributes to biofilm formation. And this information can then lead to new knowledge being found about biofilm formation in Pseudomonas aeruginosa. If you want to see more detailed information on our project, please check out our poster. Thank you for listening.